The overall goal of the following experiment is to obtain quantitative information about the activity level of PAD4 in an easy-to-reproduce assay using fluorescence. This is achieved by incubation of PAD4 with the designed substrate analog ZR-CoM. The protein is incubated either at room temperature or 37 degrees Celsius to allow for the conversion of the arginine derivative into the citrulline product, which results in a covalent modification of the substrate. Next, trypsin is added to the solution in order to generate the fluorescence signal difference. The results show the addition of active PAD4 to a solution containing the pro-fluorescence substrate leads to a covalent change based on its decreased susceptibility to trypsin and therefore the lower fluorescence signals.
The main advantage of our technique over existing techniques, such as the one that looks at the release of the volatile ammonium byproduct, is that we monitor the modification of the substrate using fluorescence. Demonstrating this technique will be Mary Sabulski and Jonathan Fura from my laboratory.
To work with PAD4, first transform PGX plasmid containing PAD4 GST fusion into chemically competent E. coli cells for PAD4 protein expression. Thaw the cells on ice. Secondly, mix the thawed cells with 1 microliter of the PGX plasmid containing the PAD4 gene in a 5 milliliter culture tube. Incubate the mixture on ice for 10 minutes and gently shake it every 2 minutes. Then, heat shock the cells at 42 degrees Celsius for 40 seconds. Immediately after, place the cell plasmid mixture back